In the last video, you learned how organelles interact to make ATP. Video 6.2 will guide you through how organelles work together to help cells make proteins. The focus will still be on the structure and function of single organelles, but also how organelles interact, and in addition, you'll learn about something called the endomembrane system. I'm excited to tell you all about that, so let's press forward. The process of making proteins is called protein synthesis and involves two basic steps: transcription and translation. Transcription happens when DNA is read in the nucleus and an RNA copy of the information is made. The RNA takes the genetic information out of the nucleus and to a ribosome. Ribosomes read the information in RNA and use that information to link together amino acids and make proteins. Now, that's called translation. Finally, the proteins are folded and delivered to wherever the proteins are needed. We'll take a closer look at this process by examining how these organelles interact. First up is the nucleus, which has a way cooler structure than you ever thought, but also a way simpler job than you ever thought. Now, structure-wise, a nucleus has its own membrane called the nuclear membrane or the nuclear envelope. There's a darkened spot in the nucleus called the nucleolus, and of course, the nucleus is where the DNA is found, and the DNA is arranged in chromosomes. The job of the nucleus is to hold, protect, and help organize the DNA, but also the nucleus is the site of ribosome synthesis. Let's match up these structures and functions. The nuclear membrane isn't just a regular old membrane, but has these specialized structures called nuclear pores. And just like a pore in your skin, a nuclear pore is a tiny little hole. The pores are large enough to let RNA out that's made during transcription, but too small to let this big old DNA molecule out of the nucleus. And this way, DNA is kept isolated in the nucleus from the rest of the cell's reactions out in the cytoplasm and other cell compartments. And this makes the DNA less likely to get damaged or mutated. Here's another view of nuclear pores that you can see them in red all over the surface of the nucleus, and they're just all over, and you can see them in an electron micrograph, a fancy high-powered microscope. Nuclear pores even have this visually stunning structure called pore complexes or rosettes. So what is this darkened nucleolus structure about? Well, the nucleolus is a specialized condensed area of genetic material responsible for making ribosomal subunits. A ribosome is made up of a small and a large subunit, both of which are partly made in the nucleolus, part of the nucleus. And those subunit pieces pass through the nuclear pores in the nuclear membrane so that the ribosomes can assemble outside of the nucleus and make proteins. But how do ribosomes work exactly? Well, there's actually two kinds of ribosomes. One type, called free ribosomes, float around in the cytoplasm. If a protein has a job in the cytoplasm, that protein actually gets made by a free ribosome. Now, the other type of ribosome is called a bound ribosome, which is bound to the rough endoplasmic reticulum, or rough ER. And if a protein has a job in another organelle or on the membrane or needs to get out of the cell and secreted to do its job, well, then that protein is made on an ER-bound ribosome. Now, regardless of the type of ribosomes, though, all ribosomes do the process of translation, which is using the RNA code to put together the amino acids in the right order to make polypeptides that can fold and become proteins. But what about this rough endoplasmic reticulum thing? The rough ER is a big membrane network. That's what reticulum means, network. The rough ER is attached to the nucleus, and some nuclear pores actually feed right into the rough ER. And attaching to the nucleus is important because what makes the rough ER rough is attached ribosomes. So here's the process. An RNA leaves the nucleus and attaches to a ribosome on the rough ER. And as a protein is made from the RNA instructions, the protein, polypeptide, enters the lumen, or center space compartment of the ER. In this center space, some magic happens. This is where proteins can get folded and even modified by attaching carbohydrates, simple sugars, to the proteins to make glycoproteins. And once these modifications are in place, then the proteins are sent to another organelle, the Golgi apparatus, for further processing. That was a lot, so let's step back and look at that another way. This poop emoji thing is a ribosome, a bound ribosome that's attached to the rough ER. And the ribosome is making a polypeptide by translation and feeding the polypeptide into the rough ER lumen. Next, the polypeptide is folded and sugar chains are added. And finally, a piece of the ER membrane literally detaches. And once that piece detaches, the bubble containing the proteins is considered a vesicle. And this vesicle is off to the Golgi apparatus. All right, so then what's the Golgi apparatus? The Golgi apparatus is another big old stack of membranes. Vesicles are received from the rough ER on one side and dump the proteins into the Golgi body. Now, when passing through the stacks of Golgi membrane, the proteins can be further modified, but the proteins are also sorted in the Golgi based on the protein's final destination. Lastly, pieces of the Golgi membrane pinch off to become transport vesicles and move proteins to their final destination. Let's zoom out a bit to put these pieces together. Now, after being made, folded, and modified in the rough ER, proteins move in vesicles to the Golgi apparatus. The Golgi apparatus further modifies proteins, packages the proteins into vesicles, and releases the vesicles to take the proteins to their final destination, which in this case is outside of the cell. The crazy thing here is that once this vesicle from the Golgi fuses with the membrane, then the membrane of that vesicle actually merges to become the membrane of the membrane. That's pretty nuts. And this whole thing of vesicles pinching off of one spot and going to another spot and fusing to that second spot is called the endomembrane system of the cell. So the endomembrane system refers to how the membrane-bound organelles can break off pieces of themselves that become vesicles, and then those vesicles go and merge onto other membrane-bound organelles. You know how ER membrane pinched off, became a vesicle, then fused to the Golgi and became part of the Golgi? That's the endomembrane system. How about when a piece of the Golgi budded off, became a vesicle, and then fused with the plasma membrane to become part of the plasma membrane? Yep, that was the endomembrane system too. Remember in that last video when a vesicle forms by budding off of the plasma membrane with the food particle inside, and then fused with the lysosome, and those became one big thing? That's part of the endomembrane system too. Wow, that was a lot of organelles, but here are some key takeaways. Don't forget our overall theme that organelle structures suit organelle functions, and that organelles interact to do complex jobs. One of those jobs is protein synthesis. And the nucleus is involved by housing DNA, the genetic information, and the nucleus is the site of ribosome synthesis and RNA transcription. Free ribosomes translate that RNA that's made in the nucleus to make cytoplasmic proteins. In contrast, ER-bound ribosomes translate RNA to make proteins that are secreted outside of the cell or proteins that are bound for other organelles. Next, the ER will modify proteins and send proteins to the Golgi for further modification, packaging, and sending to the protein's final destination. The last thing we touched on by putting this all together is to explain how membrane-bound organelles can interact with each other using vesicles, and that's called the endomembrane system.